This is part two of the Microbial Genetics lecture video. Um, in this portion of the lecture, we're going to talk about how do antibiotic resistant strains of bacteria arise and how are resistant traits transferred readily to other bacteria. And in general, we can address the answers of those two questions as well, bacteria need to change genetically. Since bacteria reproduce asexually through the process of binary fission, which is cloning, that means that all bacteria for a specific species are genetically the same. However, there are mechanisms which bacteria can change genetically. Um, there are, in general, two ways in which bacteria can adjust to changing conditions, one being gene regulation and the other being genetic change. Genetic change alters the genotype of the organism, which in turn may cause a change in the phenotype, which is the observable characteristics or the actual proteins that are expressed. And there are two mechanisms in which bacteria can change genetically: mutation or gene transfer. So a mutation is a change in the existing nucleotide sequence of a cell's DNA. So if there's a change in a cell, that's passed to the quote-unquote daughter cells. And those daughter cells are referred to as mutants. We call this vertical gene transfer since that new trait is passed vertically to the offspring. So if we have this mutation highlighted here in green, all cells from that new line from there on out would have that same mutation. So since that mutation arises in a single cell, it's passed to the next generation, vertical gene transfer. Now these mutations we're talking about are spontaneous mutations, which occur naturally in cells. They're random events. They would occur 1 in every 10 billion nucleotides during DNA replication. So think back to the first lecture video. Which enzyme is responsible for making the mistake? And there are three types of mutations that would happen during DNA replication. These are called point mutations. So there are substitutions, insertions, and deletions. Insertions and deletions cause what are referred to as frame shifts. That's because the reading frame in which the proteins are actually produced then does change. So looking at a substitution mutation, substitution is simply just a switch from one nucleotide to a different nucleotide. So it's supposed to be an adenine, and the DNA polymerase puts in a thymine, for example. So I have these random letters here, which aren't so random, but if we group them into threes, just like we would for codons, this sentence here says the cat ate elk. Okay, so this is what the cell's genome is trying to say: the cat ate elk. So if we substitute a single letter or nucleotide, we can change the meaning of that phrase. So again, assuming these are A's, T's, C's, and G's in a nucleotide sequence, if we change the C here to an R, we now change the meaning of the sentence. It's not the cat eating elk, it's the rat ate elk. Okay, and then again, if we change a random letter, substituting one letter for a different one, we make that phrase basically meaningless. Okay, so this is what can happen in DNA during DNA replication. If we have a ATCG sequence, okay, we're supposed to have an A. DNA polymerase puts in a T or a G or a C. So we substitute the one nucleotide for a different nucleotide. An insertion or deletion can cause more serious changes, which result in what we call a frame shift, like I mentioned. So that can affect the protein much more seriously. Here's that same example of the cat ate elk. If we insert a E here, so here we have we inserted the R here, sorry, not the E. But you can see why I made the mistake here, because inserting that R, we shift everything to the right. So rather than it say the sentence say the cat ate elk, we now have something completely meaningless. So that would completely change the protein sequence for that cell. A deletion here, we have a deletion of the H. And again, completely meaningless sentence. So the effect of a mutation on the protein can be either silent, missense, or nonsense. A silent is a change in the DNA resulting in a change in the mRNA, resulting in no change in the amino acid sequence. That's due to the redundancy of the genetic code. Um, just as an example, AAA in DNA is changed to AAG. So we have a substitution for that's supposed to be A, it's now a G. That means that we have a change in the mRNA. This is now UUU. This one's UUC. But UUU and UUC are both codons for phenylalanine. So there's no change in the amino acid. So the mutation has no effect, a silent effect. Missense mutation is when we have a change in the DNA resulting in a change in the mRNA resulting in a change in the amino acid. So here now we have a different amino acid change from one to another. The effect on this depends on the location in the protein sequence. So if it's a critical location, the amino acid is changed, say like the active site, that could lead to a loss of function of that enzyme. If it's in a less important location, there's potentially no adverse effects. A nonsense mutation is a change in the DNA resulting in a change in the mRNA, resulting in a stop codon. The results usually in non-functional proteins because we would get shortened proteins that wouldn't have all the amino acids needed for a functioning protein. All right, let's look at some specific examples here. Effects of mutations on proteins. This is the DNA sequence. This is the mRNA amino acid sequence. This is what we're going to be comparing the remaining slides to. So here's where our change is. We have A to U, that's normal. And sorry, where's the change here? We're going to have a change in the C here. So it was a C, now it's a T. That means that we now have an A in the mRNA. So our normal protein, we're supposed to have met, lyse, phi, gly, stop. We now have a change to a serine. So missense, we have a change in the DNA, change in the mRNA, resulting in a different amino acid. Nonsense, here we have a change in the DNA. It was a T, now it's an A. So now that we have that change in the DNA, we have the change in RNA, we now have a stop codon. So we have a shortened protein, basically it's a start-stop sequence. There is no protein that's actually produced. Looking at a second question here that we mentioned before, how are resistant traits transferred readily to other bacteria? That occurs through what we call gene transfer mechanisms. So this is when a gene is acquired from another non-related organism. So here we have a plasmid inside a bacteria. This plasmid is transferred to this other bacteria, and now we go from a horizontal to a vertical gene transfer mechanism. So this bacteria here is non-related potentially to this bacteria, but it acquires the plasmid from this bacteria, horizontal. And just remembering back to the vertical gene transfer where we had a mutation resulting in that change. Vertical gene transfer, these would be all related bacteria. Horizontal, non-related bacteria potentially. So gene transfer results in genetic variation, and genetic variation is needed for bacterial survival. There's three ways in which gene transfer in bacteria occurs. There's transformation, conjugation, and transduction. And these methods are responsible for the spread of antibiotic-resistant genes. So we have a mutation that results in antibiotic resistance, and then we have these gene transfer mechanisms that can spread these to non-related bacteria. So transformation is the uptake of naked or cell-free DNA by a competent cell. So sources would be like a lysed cell, a bacteria that dies, and then this bacteria taking in a bit of the DNA and actually incorporates that into its chromosome. So some genera of bacteria can actually secrete DNA into the environment, and other bacteria can pick it up. But the cell has to be competent, meaning it has to be able to um, pick up DNA. Some bacteria can do this naturally, others do not. So the first evidence for transformation occurred through these experiments by Frederick Griffith in 1928. What he did is he used two strains of Streptococcus pneumoniae. One was a virulent strain that causes pneumonia, and the other was a non-virulent strain that doesn't cause disease. So the virulent strain produces a capsule, the avirulent strain, no capsule, no disease. And what he did is he took the living bad bacteria, the encapsulated one, injects it in the mouse gets sick, and the mouse dies. And if you remember Koch's postulates, you must be able to culture that from the organism from the infected and grow it in the lab. And that's what he did. He was able to inject that into the mouse got sick and died, and then regrow it in the lab. He then injected the non-encapsulated bacteria, so the one that does not cause disease, injects that into the mouse remains healthy, and he does culture that same organism from the mouse. Now what he does is he takes the heat-killed, so number one here, heat-killed bacteria, injects it in the mouse remains healthy, and does not culture anything from the mouse, which makes sense. If you kill the bacteria, you're not going to have any growth. Now what he does is he's going to mix the heat-killed and the avirulent strain. He's going to mix it together. So he mixes those together, the non-encapsulated and the heat-killed, injects it into the mouse gets sick and dies. And what he does is he cultures that mouse and he finds the encapsulated bacteria. So what happened was we have the non-pathogenic strain and DNA from the heat-killed virulent strain basically are mixed together, so some genes here were taken in by the non-pathogenic strain. This is incorporated into that bacterial's genome, and that transformed bacteria is now pathogenic. So this bacteria picked up a new trait and was which allowed that bacteria to produce the capsule and make the mouse sick. Conjugation is another gene transfer mechanism. This is where one bacteria passes some DNA, often in a plasmid, to another bacteria using sex pilus. So it can either be a plasmid or short pieces of chromosomal DNA that can be transferred through the sex pilus, and this now the new bacteria would have a new trait. And then transduction is the last one, where bacterial DNA is transferred from one bacteria to another by a virus. And this is done through viruses specific for bacteria, referred to as bacteriophage. The reason why viruses are able to do this, it's a mistake during viral assembly. We'll talk about this in chapter 12 in the virus chapter. So, looking back at these questions, how do multiple resistant strains arise and evolve, or how do antibiotic resistant strains arise? The answer to that is mutations. Okay, some bacteria are naturally resistant to antibiotics, but if you're not naturally resistant, if you're going to become resistant, usually we have to have a mutation resulting in a new trait. Those genes can then be transferred to other bacteria through the three gene transfer mechanisms: transformation, conjugation, and transduction. So another question here, why must bacteria rely on these methods for adaptation and survival? And that's because bacteria reproduce asexually. In order to have genetic change for evolution, you have to have mutation and you have to have gene transfer. So these bacteria have basically develop new traits. This will conclude the Chapter 9 Part 2 video. Make sure you complete the study guide and the e-learning quizzes